Harrington. Welcome. Thank you for being here. I'll give you the clicker as well.
Great. Thank you.
Have fun.
Thank you very much for the invitation. Can you all hear me? Yeah, great. Thank you very much for the invitation to be here and to tell you about some of the work that we've been doing.
Heather, could you hold the microphone a bit higher than
Sure.
Yeah, thanks so much.
Great. So I wanted to motivate some of our research is starting with some type of complex biological system. And we're really working across molecular cell biology and genetics, and more linking with medicine and the clinic. So my title slide, you can see the picture here is...
Move it down and move it up and down a little bit.
Okay, great, thank you. You can see that on the left here, this is a tissue sample from cancer, and on the right is what I guess it's a visualization of a quantification of such a network of such cell spatial organization. And what's so exciting about the methods and algorithms that we've been developing is that they are interpretable and now computable, which is very different than they were even 10 years ago in these fields of computational mathematics. So this kind of is crossing many different scales in biology. So in our bodies right now, we have trillions of cells. And in every cell, we have the same genetic makeup, right? Yet, there are different proteins that are expressed. And so we heard earlier that from Neil that looking at how do you predict protein folding. So going from the genome to proteins to then interactions of many proteins to then looking at many different types of cells. And each of these scales in biology are really complicated, and we don't understand how it works, and we were really trying to understand the spatial organization and the shape of this biological data. And right now I'm just showing you a picture, it's static, but all of this is very much dynamic. And so we can add that onto the challenges we have in these types of data. So the work, I'm going to give you kind of a sample of different biological problems we've looked at, and each one has required us to develop new algorithms which are now computable. So some of the tools that I'm going to talk about weren't computable before. So this is very much joint work with many different collaborators. Many of them are now, that I have here, are in Oxford, but many other places. And this is really ranging, as I said, from very theoretical mathematics, proving and developing a new invariant, all the way to the computation of the algorithms, the computation, the implementation, the application to the clinic, right? And so being able to cross all of that, I think is very special. We have this Center for Topological Data Analysis in Oxford, and it's really application-driven, which is very different than many areas of computational mathematics. This is not necessarily starting with an application. So today, I'm going to tell you about one algorithm, and it's called persistent homology. And what's so special about this algorithm is that we can quantify the connectivity of data in different in different ways, all in one descriptor. So this is crossing different scales of your data. So often when in data science, you pick some threshold for your data and that's it. You only study at that threshold. We really want to cross all the different thresholds. We also want to cross different biological scales. As I said, we're able to interpret the features in the data so we can go back to the application domain expert and say, this is why we're finding, or this is the interesting area in the data that our descriptor is capturing. What's so special is that persistent homology has these stability theorems, which means that if your data moves just a little bit, you still get the same invariant just a little bit. And that's really special to say this is provable. within some small perturbation to the data. And then in the last 5 to 10 years, algorithms have been developed to be able to integrate this with many different machine learning and vectorization methods so that we can study, instead of just one data set, many and comparing them. So standard statistical data analysis, which wasn't possible, or at least developed before, and I'll explain why in a minute. So my motivation slide, right at the very beginning, showed cells, a bunch of cells. And let's pretend we have these cells, for example, immune cells in a cancer, and we all know that, whether you want to have immunotherapy or some other type of therapy really depends. A pathologist will look and visually see where these immune cells are. Okay, so let's pretend that our data, I'm just looking at points in R2, with we can use Euclidean distance, but you have some metric, and for example, pretend these are our immune cells on the left, and inside is the tumor. Okay, we want to study this. Now maybe it would be a perfect ring, but it's a noisy ring and you have points that are sampled for that snapshot of when you got the tissue sample. And what we would like to do is, you can't see it very well, but instead of clustering these points, we can really see that there's points around a ring. So what persistent homology does is you build a sequence of nested simplicial complexes. These are higher order networks. So you're building, so you say, okay, let me look at some distance and increase the distance around, so thicken the points, and any points that are intersecting, then we build, for example, an edge or a triangle, and you can fill these in, a tetrahedra, etc. So you build this nested sequence of spaces as you increase the epsilon balls around them, and then you get to a certain epsilon. So if we look along the x-axis, we get to a certain epsilon where the points will all link up, and we have this feature which this looks at our topological invariant in different dimensions. And this feature here shows that there is a persistent loop in our data. Okay, and so what's very nice is that now we have, it's all algebra under the hood, but what we have here is a visualization we call barcode, where the left endpoint and the right endpoint. The left endpoint is, for example, we have 20 points, and the right endpoint is where they merge. So these are the number of components or clusters as you increase epsilon along the x-axis. And this is just a multi-set of intervals. So x, y points, right? It's a multi-set. And if you say, okay, let me do some statistics, what does that mean? It's a multi-set of intervals, right? You don't have, it's not some nice space to do some statistics. And that's kind of the next step of what a number in the community have worked in topological data analysis. So we have a descriptor here called a barcode that describes the number of connected components, but also the number of loops or the number of voids, etc., in higher dimensions. So this is a descriptor of your data, and you can think of it as some type of dimensionality reduction, where I can interpret and say the features here. Okay, and then we can continue along this epsilon until everything is filled in, you get a complete complex. Okay, and as I said, so we have this pipeline where we start with our point cloud data, for example, and you continue to thicken the points and you connect the dots and you build this nested sequence of simplicial complexes of these graphs, and then you compute these topological invariants that describe how they're connected, and we output, underlying everything is algebraic structures, but what we output is a barcode, and it shows where features are born and where they die. For example, here was the one persistent loop that we can see. Okay, so we wrote a roadmap to this computation of persistent homology 6 years ago, and what's remarkable is the field has changed so much. So this looks very simple, right? I just showed you something in R2. You can look at any high dimensional data, but we couldn't compute this. So the standard algorithms, we couldn't compute any of this with real data. We were running the supercomputers in Oxford, and we bought bigger supercomputers to actually run things. So it was really computationally intractable. And in 6 years now, you can run things on your laptop. So the field has massively changed, and it has in part because a larger community became involved that were experts in numerical analysis. And so this, the standard algorithm scales in the number of simplices, these triangles, and cubed. So it really could be, at the worst case, very bad. And now it's almost linear for one parameter persistent homology. So this is only one parameter. This is where epsilon, we're looking at one scale parameter. So this is vanilla, the simplest. Has anybody heard of persistent homology here? Okay, a couple people, but I'm really happy to hear that I've introduced this new area, right? Everybody's heard of ChatGPT, but now you'll go home and say, oh, I learned about persistent homology that gives me shape, a quantitative way of capturing shape in the data. Okay, so that's kind of the start. This is my summary of this is what persistent homology is, and now I want to give you a flavor of the different directions that we're going in, different ways that you can apply this persistent homology algorithm. It's not just the point cloud data that I showed. It's really versatile. So you can use it with undirected networks, weighted networks. So examples are in fMRI. Neurotopology is a very large field now. The Blue Brain Project has used it. Many neuroscientists are using these types of algorithms because you look at fMRI networks, and these are weighted networks, and so they're using that here. Grayscale digital images, that's for the single parameter persistent homology, and it's a little bit more complicated because you build slightly different complexes, these weighted, but they're very, it's very flexible is what I wanted this slide to show you. And then finally, to be able to use it and apply it to other areas is being able to interpret what we're getting out. So as I said, this multi-set of intervals is not very helpful, but what you can do is then build a map. So we build a function, a sequence of functions where we can then look at this into some suitable Hilbert space, and there's different ways to do this. That were only recently introduced, right? So this is relatively new and has been successful. So you'll, I think in my talk today, I'm going to use both persistence landscapes, which is where you basically are building like a tent function on each of these bars. And then you can see at any value along this epsilon, what is the most persistent. You can see by the height, and that is given, for example, the most persistent landscape. So these are statistical ways you can take averages and put into some machine learning pipeline. Persistence images is another great way that we've been using this to vectorize the barcodes. Okay. Then there's other kernel-based methods. I mean, but really there's a recent review this year that kind of goes through, and this was, I think there were like 8 authors, but Vidit Nanda and was one of my colleagues and one of the authors there, and they really go through all the different vectorization and machine learning ways to integrate persistent homology. Okay, so now that I've kind of given you an overview of what is persistent homology, the vanilla version of it, I'm now going to tell you how we've been applying this and really kind of innovating extensions to these pipelines. The first one is with experimental data. So when I say the word model, most of you in the room probably think of a machine learning model. Maybe. It's a computing conference. So, but there's many different types of models. There's, and this is why communication even within different within an area is so important that, for example, this is a mouse model or a mechanistic model or a protein like structural model or a PDE or agent-based model, right? And so I'm going to touch just briefly on how these are different types of data and we've been using them. And what's, I think, nice is most of the examples I'm giving you are very low dimensional in some way, so you can see it, but we can apply it to higher dimensional data, and there's some theorems as well for what you can do there. And the point is each of these applications, because it's not standard use out of the box, we kind of have to adapt things. And what's changing as well is it's getting more versatile. So more of the software now in implementations, you can kind of pick it up and use it, which I was shocked, right? You can go online and within an hour you're using persistent homology, which was not the case to even get things to build before. So here's an example from cancer, which is the growth of blood vessels towards a tumor. And it's a very complicated process, and we don't understand it. But they know that the shape of the blood vessels matter a lot, right? So if you have a tumor and there's a lot of blood vessels that are able to bring it brings oxygen very well, then you might want a drug that goes like chemotherapy, that's actually going to go through the blood and kill the tumor. Whereas if the oxygen in the blood vessels are very complicated and there's not that much, it's a bit less vascular, surgery might be better in some way, right? So this was the motivation, at least when we learned about this problem, is how do we quantify the vasculature near a tumor? And these are kind of all of the different molecules and proteins involved that they can measure, as well as the structural data. And so we developed as I said, you have some different type of data, and we develop a way to look at this nested sequence, this lens, this nested sequence of spaces, and then get some output, some vectorization. And we did this with vascular networks, but first we had to develop which is the appropriate filtration. And we looked at inspiration from others in the topological data analysis community. So one was looking at brain artery trees, and they did this sweeping filtration going from the brainstem up. And we thought, okay, that's how we can continue to add this nested sequence of spaces. There's a direction there. And then we thought, but that doesn't quite work with a tumor, because there's no real clear direction of the vasculature. And then we, colleagues in Southampton, were looking at COPD in the lungs, and they have, again, some natural direction for how to filter the data, but we didn't really have that. And then colleagues at EPFL were looking at neuron morphology, and their point was they have a root in the neuron, and they could filter up. So we thought, okay, well, maybe we can do the filtering up radially, kind of how they are doing it in the neurons. And that's exactly what we did. With different types of microscopy data, so we looked at multiple types of experimental technologies and working with different labs and different treatments, we get this three-dimensional data. So that was, somebody's entire PhD project was reconstructing this three-dimensional data. And then they were actually part of the AlphaFold project after this. And so that we used their algorithm and then built this filtration radially, so as we add it. And what was so wonderful is that for each of these, whether we look at connected components or we look at the number of the dimension 1 topological components or dimension 2 topological components, we could then interpret what it means. So, for example, how bendy, the tortuosity, the number of loops, which of course you would think a vascular system doesn't have loops, right? I mean, that's not very efficient. In a tumor, it does. So we could capture the loops and also the voids, the avascular regions. And we applied it to with different experimental labs and different types of microscopy and different types of data and different experimental conditions. And we were able to find that across all of these, we found that if they added some agent that decreases the vasculature, and we could quantify that the loops are decreasing. And a similar thing with the voids. So this is experimental data, but of course we want to understand why this is happening. What are the factors? And these are mechanistic models. So I mentioned that. And there are hundreds of these models. And in the UK, it's one of the strongest places for looking at models of angiogenesis, this new vasculature near tumors. So what we can take is our modeling parameters, this computational framework, with the models of tumor-induced angiogenesis, and we can generate a lot of synthetic data, where we're looking at the growth of blood vessels where you might have a tumor on the right, and the initialization of the vessels on the left. And we can use persistent homology, this computational tool, to then study, so we can use standard homology, which is a standard invariant. We don't get that much information. We can use persistent homology, where we're tracking along the X space, and we can get the number of loops and locations, and then we can build on this. So we had one of the first applications of building on this in what's called extended persistent homology, where we can then group according to the topological features in our parameter space along the x and the y, and then here are representatives of each of those clusters. So you can see that the groupings definitely distinguish different dynamic processes. And then we might want to ask the reverse question, given some data, can we go backwards? And we built this topological Bayesian framework, which I think in the interest of time, I'm going to go kind of quickly, but the idea here is that we can not only capture the number of loops, the location of them, but also their size. And so this was nice because we can then go back to the domain experts and show them and explain what the differences are. And our framework works very well for from the observed data to the posterior predictive. Okay. Now I've showed you persistent homology. I've shown you a couple examples of how it's useful in one parameter, but now we want to be able to interpret. And so remember, I'm going to give you the example first, and then I'll show you. So the motivation in this problem was the structural biology data. So if we have protein structure, we want to understand what are called homologous proteins. So these are proteins with almost the same structure, but they have different shape. Okay? Or they can have. And we looked at knotted proteins. So there's a small class of knotted proteins. And there are some standard ways that people look at this, which is called sequence homology representative. And there's also where for these knotted proteins, whether it has a deep or a shallow knot. And we wanted to ask, can persistent homology help us here? So again, we're using the same framework as before. We take the points, the backbone of these carbon atoms in the protein, and we grow balls of some epsilon around each point. Then we compute the topological invariance, and we can get a barcode, or the x, y can be plotted as the birth on the x, and the y is the death. And so this is an equivalent visualization of what I've been showing you with barcodes. And what's really cool is in the last couple of years, now we can go back and say, all right, this point in the diagram, this bar in my barcode, what were the points in my data set? Which sounds really trivial to say, but ends up being a difficult integer programming problem that you can't do for all dimensions, but you can solve in certain cases. And so we then we compute these points in the original data set that are involved in some descriptor we have. Okay, and then we can also do statistics. So we can look at the global analysis. We can look at families of these proteins, and we see that they, persistent homology has a different average family for different families of these, different groupings of these proteins that are known to be similar. So that's good. It's kind of a validation. But then we asked, can we look at the knotted and unknotted of these homologous proteins? And what we see is that the topological summary we get out has this kink. Can everyone see the little kink in the right here on the knotted? I don't know the pointer. I'm struggling here with the pointer. So you can see this here. And what we found is that the difference, that little piece there, it actually, with the generators, it identified that there was this crossing change. So it localized that. So we can look at the global structure of a family of proteins, but we can also look very locally at what's happening structurally and interpret it, and it pulls it right out for us, which I think is very powerful here. Okay, and then the final thing that I wanted to talk about is extensions to this pipeline. So I've talked about one parameter. I've talked about how we can interpret it with some of the generators actually going back into the data. But now we can then use these generators to look at not just one generator, but multiple generators. Right? So instead of looking at, you have your points in your, for example, protein or some other trajectory, we can take each of these points, these persistence points off the diagonal here, which would be long bars in the barcode, and we can go back and say what were the loops in the data in the generators. And we applied this to a number of different problems. So we take our point cloud, we compute persistent homology, then we say, okay, these are the red, for example, the red show which points were involved with this one feature, and green are the data points in the original data involved with this long persistent feature. And we do this. And then we analyze that. So we can analyze it with hypergraph, which again, in the last 2 years, these are very new hypergraph network techniques that we can then say, okay, these points in your data are really important, and they're not important in the standard clustering. They're important in these higher order connectivity and higher order features. And then it's been really cool because we've applied it to many different methods that have surprised us. So we're collaborating, getting up earlier, staying up late with some experts in Australia, looking at this with chromatin, which again is a very complicated, it's crucial for in biology, but the structure of chromatin at different scales is really challenging to quantify, and so some of these ideas are really useful there. And so this is just an example of where we can find some interpretation that some of the domain experts are very happy that these hypergraph network measures are useful. Okay, so then there's some details which I think I will skip just in the interest of time. Did I start a little late, maybe?
You started a little bit late. Went for about half an hour, is that correct? Half an hour to 40 minutes for stories, so if we could leave at least 5 minutes for questions and finish
Great,
around
okay,
11 would be great.
I've been going for 20 minutes, or is that right?
Yeah, I believe so.
Okay,
Yeah.
great. Okay, so then I just wanted to say that, yeah, so generators, of course, you could imagine that the points that we select to create a loop could be slightly different. And so this is not a unique thing mathematically, but we can actually go through and show that in the case for these types of data, it is fairly stable, and there's multiple ways to compute the generators, and it is still stable. And we've applied it to other types of models, and again, we can see that for different classes of random, these diffusion models, we can predict based on the trajectory, this hyper-TDA can project which of the appropriate models that the data came from fairly well. Better than, I mean, this was a challenge, and our results are comparable with some of the top results. And so the final thing is just where we started with the spatial locations of cells, is we want to understand patterns. And so almost everything I've talked about today is very much biology, but we can look at other types of patterns. This was not intentional that currently in the news we're thinking about deep sea spatial, I wrote this talk before, but we were looking at coral, algae, and turf, and we're interested in the spatial changes, and we have data of how it's changed over time. And so in certain quadrants, we have whether something's been coral changed. And we want to quantify that. And there's also mathematical models to do this. And the challenge is we're looking at now dynamic data. And so there's new approaches to be able to study the topological connectivity over time. And so I'm not going to go through too many of the details, but instead of looking at a single snapshot of the data, we can do this over time, and we no longer have a nested sequence, right? Our data is no longer just inclusions and building. You have things that are changing, and so you have to build a slightly different lens, this filtration. Okay, so we've done this and been able to identify, for example, bifurcations in the model, and we can distinguish different grazing rates of the fish that involves whether something becomes all coral or all algae. And you can also think about this looking at this complex system of the tumor microenvironment. Again, this is looking at the immune cells, and so I'm ending where I started, the immune cells on the outside of a tumor. And as they kind of move in, you can think about this on mechanistic type modeling or actual pathology modeling. And this is an area that we've been focusing on a lot with the Cancer Institute in Oxford. I think for the interest of time, I'll go kind of quickly through kind of the extensions, which are looking again at the connectivity and the shapes that we get, these higher order shapes. But what arose in this, when we were doing this analysis, is that the data is not just a little noisy, but you have true outliers. So there's artifacts in the data. And you can imagine if I gave you at the very beginning of my first slide of the ring, I put a point in the middle, you no longer see the ring structure, right? And so to be able to, so this is single parameter, and then when you go to multi-parameter persistent homology, you need, there does not exist a barcode that's analogous when you try to do this with for example, density and your space. So we have to look at new ways. So this is, for example, a codensity and a distance by filtration instead of a single parameter filtration. And the mathematics becomes a little bit more complicated, and certain things are not possible. But despite that, we can get incomplete invariants that describe this, and then build, these statistical methods on top of it. And so this is something one of the students did, was building these multi-parameter persistent homology landscapes, which we then applied to this cancer immune cell locations in cancer data. And I think what's really important and exciting is that this is not just kind of going backwards and saying, can you explain my data? It's also saying, can you make predictions, which is ultimately what we all want to do, right? That's why we have models. We want to make predictions. And what we predicted, so we could explain something that the pathologist could validate and see and is statistically significant, but then we can also make some predictions, which is we had the cell density, but we also had in this data, very special, was the cell oxygen level. And we make a prediction, which we need to test still, which is that the oxygen level, which is really difficult to measure. Of course, we all know oxygen levels is super important in all cell functions. The different oxygen levels appear to be, this codensity, the density parameter, seems to be a good proxy for the oxygen level. And that's something that the clinicians were very excited, and so we're getting data from Manchester to study this. Okay, so I would like to thank you for learning a little bit about computational topology with me today. This is just one of many different types of areas in computational mathematics. I think in Neil's talk earlier, there was a touch about quantum computing and where things can be really useful. There is a quantum algorithm for persistent homology. And I think complex biological problems, there were a couple examples of biology, are really a great test bed for where you can go and where algorithms are really needed to understand at such small scales and very high dimensions. So thank you very much.
Thank you, Heather Harrington.